Hello everyone. Today I will tell you something more about functional insights in human milk oligosaccharides, or HMOs. These are key early life bioactives. I'm Hanne Tytgat and I work at Nestlé Research in the Institute of Health Sciences in Lausanne, Switzerland. Let's dive in. Early life offers a critical window to lifelong health because it's in this time period that the microbiome and the immune system are maturated. Indeed, when an infant is born, its gastrointestinal microbiome is sterile. It means that during the birth, it receives the first microbes from its mom, mainly Enterobacteriaceae. Over the next few weeks and months, the gut becomes increasingly anaerobic, and we see that more species start to pop up. These are mainly bifidobacteria, which is largely steered also via breastfeeding. This bifidobacterial dominance is really key to early life and is a typical feature of an early life microbiome. Upon the introduction of more diverse complementary foods, so diversification of the diet, we also see a rapid diversification of the microbiome. We see more adult-type species popping up, which further a trend that is further established during the following months and years, until a mature microbiome is established, which is highly diverse and resilient. This will offer a strong basis for lifelong health and help the infant to combat infections and to recover from dysbiotic events. The increase in microbiome diversity over the course of early life goes hand in hand with also the training of the immune system. The immune system really is trained during this critical window, where it learns to distinguish between good bacteria and bad bacteria. It learns to distinguish pathogens and to then react to prevent infections. So microbiome maturation goes hand in hand with the maturation of the immune system. And beyond that, a healthy microbiome is also important to strengthen gut barrier function, also has a role in modulating the developing nervous system. You might have heard about the gut-brain axis, where we see that there is a link between the microbiome and the brain, affecting, for instance, mood or some disorders affecting the brain, and so on and so on. Evidence on this is still further developing. What we also see is that there's a strong link with the developing immune system, where the microbiome helps to educate it, as I mentioned before. Also, a good resilient microbiome helps in resisting colonization from pathogens. This means that pathogens have a harder time to establish themselves in a resilient microbiome. So really the gut microbiome development and in a strong way is linked to long-term health. But this early life is a critical window, but it's also a very cumbersome time because various factors affect this gut microbiome development. There are many risk factors that threaten the establishment of a healthy microbiome. For instance, the gestational age, the mode of delivery, the feeding mode, and antibiotic use might all influence gut microbiome development in early life. To give an example, we see, for instance, from research that C-section delivery and antibiotic use in early life affect gut microbiome development. When we see here, for instance, in gray here, we see this typical bifidobacterial peak in relative abundance in early life. We see that this really up till 3 months, which is typically around the time where complementary foods are often introduced, that the bifidobacteria dominate the microbiome of the infant. Then we see a rapid decline upon the introduction of complementary foods. All normal events. However, when we see that the infant is born via C-section, here depicted in red, we see that there is a delay in bifidobacterial dominance, and also that the relative abundance of the bifidobacteria does not reach the same height as in a normal trajectory. When we look at the trajectory for infants receiving antibiotics in early life, we see even a worse trajectory where often the bifidobacterial dominance is completely obliterated and not happening anymore. This has really strong effects on health conditions in later life. For instance, in infants delivered via C-section or receiving antibiotics, we see an increased incidence in low respiratory tract infections, a larger incidence of allergic outcomes, like for instance, asthma, food allergies, and allergic rhinitis, and a higher chance to develop overweight and obesity in later life. Many of these risk factors, for instance, C-section delivery and antibiotic use, come together when an infant is born prematurely. Another factor strongly influencing microbiome development is diet. We know that breastfeeding stimulates gut microbiome development. Because here depicted in pink, we see that the alpha diversity of breastfed infants gradually increases over time, over the months. When we look at the abundance of bifidobacteria, then we see that over the first 3 months, there's a strong increase in bifidobacterial relative abundance, which is then goes down after introduction of complementary foods. In blue, we see the trajectory for an infant-fed formula based on cow milk. There we see that there is a very rapid increase of the alpha diversity of the microbiome. We also see when we look at the bifidobacteria that there is not this typical peak in relative abundance of the bifidobacteria compared to other species, which is in line with the rapid diversification of the microbiota. So we see a very rapid introduction of several species and not this gradual increase like we see in breastfeeding. So we see that breastfeeding, or feeding mode in general, really strongly drives microbiome maturation. When we then look at breastfeeding, which is the golden standard for infant nutrition, we see that this comes from the fact that it not only has all the feed to feed the infant, but also its microbiome. Breast milk is full of nutritive components like lactose, proteins, and fat that are digested and have a nutritive value to the infant. These support healthy growth and development. But there are also many other bioactive components in breast milk. For instance, cytokines, antimicrobial proteins, antigens, and so on. Key of these bioactive components are HMOs, or human milk oligosaccharides. These are non-digested, don't really have a nutritive value to the infant, but they support the microbiome and immune function. They will support immunity, gut health, brain development, and learning. So they're really key to the infant's development. Looking further at these human milk oligosaccharides, we see that they're the third largest solid component in mother's milk. They range up to 5 to 15 grams per liter. Over 160 different structures are described, with an estimated 20 of them that represent over 80% of the total HMO mass. Most common HMOs found in mother's milk are not, or only to a minor extent, present in farmed animal milk. They can be subdivided in 3 groups. All are basically elongations of the milk sugar lactose, and if this lactose is elongated by a fucose moiety, we have the class of the neutral fucosylated HMOs, like for instance 3-fucosylactose, 2-FL, LDFT, and so on. Or with the second class are the acidic ones, where acylic acid is added to the lactose, resulting in 3-silactose, 6-SL, and so on. Or more neutral moieties are added, like for instance, glucose, galactose, glucnac. And then we have these LNT, LNNT. These can be then further elongated to form more complex HMOs of the different classes. There's not only a strong structural diversity of the HMOs, there are also a large functional aspects that are linked to HMOs. HMOs are found to support microbiome development, as I showed you before, where they can have a kind of a prebiotic effect, but they also can have a direct antimicrobial effect by, for instance, preventing the colonization by different pathogens. They strengthen the gut barrier function, modulate the developing nervous system, and have an influence on cognitive development, and they also play a role in immune competence development. Taking now a look at our clinical understanding of these functionalities, we can benefit from the fact that HMOs, since recent, also can be produced in a technical setting. Technical advances have allowed for the industrial production of HMOs, which allows us to evaluate their functional aspects in randomized controlled trials. First, we evaluated 2-fucosyl-lactose, a neutral fucosylated HMO, which was then later complemented with a neutral non-fucosylated HMO, leading to 2 HMOs. And currently, we are running a trial in which we evaluate functional aspects of 5 HMOs, where all 3 structural main groups of HMOs are represented. But let's start at the beginning. What we learned from a clinical trial where infants received formula supplemented with 2-fucosyl-lactose, we saw that this resulted in a lower relative risk of an infant to have infections compared to infants receiving a control without a control formula that had no HMO supplemented. This effect was sustained in an endeavor where we looked at 2 HMOs. Infants receiving a formula supplemented with 2 HMOs, 2FL and LNNT, we had lower effect of upper respiratory tract episodes, and also they had a lower risk to have these respiratory tract infections when they received the infant formula supplemented with the 2 HMOs compared to control formula. Looking at other effects of this 2 HMO formula showed that, for instance, infants receiving 2 HMO supplemented formula had a lower risk to require antipyretics during their first months of life, and also that this reduced the risk to require antibiotics during their first year of life. Also, during their first year of life, the infants receiving infant formula supplemented with 2 HMOs had a lower risk to experience lower respiratory tract infections, as depicted in the 2 graphs here below. Looking more in what could explain this, we looked at the bifidobacterial abundance. As I showed you before, this really is important in early life, that there's a peak in bifidobacterial relative abundance during the first 3 months of age. Here we see indeed that in the breastfeeding arm, that there is a strong dominance of bifidobacteria, irrespective if the infant was born via cesarean or vaginally born. In infants receiving a control formula, not supplemented with HMOs, we saw that this bifidobacterial relative abundance was lower in cesarean-born infants. What then we saw in infants receiving the formula supplemented with 2 HMOs is that this 2 HMO supplementation could increase the bifidobacterial relative abundance so that it could increase this, bypass this effect of C-section on this bifidobacterial abundance in early life. Also, in vaginally born infants, there was an effect on a relative abundance of bifidobacteria in the test formula supplemented with the 2 HMOs. When looking at the microbiota composition in general, we see that in breastfeeding infants, fecal community type B is the most abundant one, here depicted in red. Compared to infants receiving formula without HMOs, the control group, we saw that their fecal community type C was the most abundant. Now, in infants receiving formula supplemented with 2 HMOs, we saw a strong increase in fecal community type B. showing that the microbiota community of these infants moved closer to the one of breastfed infants. When comparing the antibiotic use per fecal community type, we saw that infants falling into this fecal community type B had a lower chance of requiring antibiotics over their first year of life compared to infants having a fecal community type C. In conclusion, this shows that the HMOs had a bigger effect in the case of dysbiosis, for instance, with the C-section-born infants. Moreover, HMO feeding drives the microbiome towards that of breastfed infants. And this microbiota community shift in the infant's fed formula with 2 HMOs relates to the antibiotic use over their first year of life. Currently, we are evaluating a randomized clinical controlled trial in which we evaluate the effects of an infant formula supplemented with not 2, but 5 HMOs. Here, also acidic silylated HMOs are supplemented, so covering all 3 big groups of HMOs. The purpose of this trial is to evaluate the tolerance and safety of this 5 HMO supplemented formula, as well as its effect on growth and the effect on gut health and immunity markers. The trial was set up as a multi-center, randomized, controlled, double-blind study of healthy term infants in Hungary, Bulgaria, and Poland. The trial also consists out of a non-randomized breastfed reference group. Infants receiving formula either received control formula, which consists out of intact protein consisting of 70% whey, 30% casein ratio, and 100% lactose, which is replaced in the test groups by either 1.5 gram per liter of or 5 HMO blend or 2.5 gram per liter of the 5 HMO blends, meaning that this is test group 1 and test group 2. So there are 4 arms in this study. Looking at the effect on growth, we saw that 5 HMO in a supplemented formula supports age-appropriate growth. The growth in these groups was non-inferior, and the weight for age and length for age z-scores were all similar between both test groups and the control, and tracked closely to the median of WHO growth standards. Moreover, the infant formula was well tolerated and supported a soft stooling pattern. When looking at the microbiota composition, we saw that at baseline, the breastfeeding group differentiates from the 3 formula-fed groups, but that over the time and at 3 months of age, we see that the both groups that received infant formula supplemented with 5 HMOs moved closer to the breastfeeding arm in a means of better diversity of their microbiome compared to the control group that did not receive 5 HMOs in their formula. This was also reflected in the abundance of bifidobacteria. Bifidobacteria relative abundance of especially test group 1 approached more breastfeeding abundance. When looking at a specific species of bifidobacteria, Bifidobacterium longum subspecies infantis, which is a known HMO metabolizing strain, we saw that the relative abundance of this strain increased and moved closer to breastfeeding compared to the control group. Also, the relative abundance of other taxa was significantly changed in the test groups compared to control, and all moved closer to breastfed infants. When looking at a pathogen, Clostridioides difficile, which is known to cause diarrhea in infants and children, we saw that the relative abundance and prevalence of toxigenic C. diff was lower in the test groups compared to the control. It moved closer to breastfeeding, and while in breastfeeding, both in relative abundance and prevalence, the C. diff, the toxigenic C. diff, was non-detectable. Between the test groups, there was no significant difference between both the prevalence and relative abundance of this enterotoxigenic C. diff. Also, in manners of gut ecology, we saw that the infant formula supplemented with the 5-HMO moved the gut ecology closer to the one of breastfeeding, of the breastfeeding arm, where we saw an increase in acetate, a decrease in butyrate, an increase in lactate, and a decrease in the fecal pH. All markers showing that gut ecology moved towards the one of breastfed infants. Also, it was found that 5-HMOs support gut functional maturation. There, two markers are of importance. One is gut immunity marker SIgA. SIgA is an important gut gatekeeping molecule. It protects against toxins, viruses, and pathogens, and is a marker of immune response. We know that in breastfed infants, the SIgA is extremely high, because there it is also influenced by other factors in the breast milk. What we saw in our 5-HMO supplemented formula-fed infants is that they had a higher SIgA levels, which were significantly higher when compared to the control and moving in the direction of breastfed. Another important marker is the gut permeability marker alpha antitrypsin, which the higher it is, the more permeable the gut. So we saw that in the test groups, there was a significant lower amount of alpha antitrypsin marked, which is a marker of a better gut barrier function in the test groups. In conclusion, I showed you that 5-HMO supplemented formula has been clinically shown to shape the gut microbiota and support an overall improved gastrointestinal environment. By shifting several bacterial taxa, including bifidobacteria, as well as changing the gut ecology towards the one observed in breastfed infants. Also, the test groups had a lower prevalence and abundance of the pathogen associated to diarrhea, enterotoxigenic Clostridioides difficile. We also saw in the test groups an increase in fecal SIgA, which is an important marker of immune response, moving closer to breastfed infants. Lastly, we saw a positive effect on gut health markers by the decrease in a marker of intestinal permeability, alpha antitrypsin, which suggests that HMOs support healthy gut function and maturation. To end, I want to share some take-home messages of my talk. The early life is really a critical window for development, where we see that the gut microbiome and immune system are established and matured. This resilient microbiome really forms the basis for a long-term health by reducing vulnerability to infection and disease. Many factors influence this gut microbiome development in early life, like, for instance, delivery mode, antibiotic use, and infant feeding. And it's there that breast milk really contains all nutrients and bioactive components to drive optimal development of the infant and its microbiome. Especially HMOs, or human milk oligosaccharides, nurture an age-appropriate microbiome. And I showed you data where it was clinically shown that HMOs support gut immune and barrier function, likely linking their effects, likely linking this to their protective effects from infection. I want to thank you very much for your attention,